Natural proteins, such as hormones and cytokines, are the body's primary tools for regulation, making them promising candidates for treating human disease. However, these proteins face a major physiological hurdle: the human kidney. Small globular proteins with a molecular weight under 70 kilodaltons are rapidly filtered out of the blood. This means natural proteins often vanish from the bloodstream too quickly to be effective, unless administered in constant high-volume doses. To bypass this clearance, scientists developed the fusion protein. By taking the genes that code for two entirely unrelated proteins and splicing them together, they synthesize a single continuous polypeptide chain. This unnatural molecule possesses the functional properties of both parent proteins simultaneously. This chart of global sales proves the success of these engineered biobetters. Since the first approval in 1998, we have seen fusion proteins like Eylea and Trulicity reach annual revenues of $9.2 and $6.5 billion, respectively, illustrating the massive scale of this pharmaceutical class. We are no longer limited to the specific molecular structures found in nature. By engineering fusion proteins from scratch, we can extend the circulation time of medicines and bypass the native clearance thresholds of the human body. Simply joining two functional domains is rarely enough. If the proteins are too close together, they can physically block each other, preventing them from folding correctly or reaching their cellular targets. The engineering solution is a linker. Designers choose between flexible linkers, often repeating chains of glycine and serine, which allow for movement, and rigid helical linkers that act like a stiff strut to force the protein segments apart. The ratio of flexibility to rigidity is precise. If the linker allows too much movement, the protein suffers from steric hindrance, essentially tangling itself and becoming functionally inert. A more advanced design is the cleavable linker. As shown in panel D, these are severed by a specific protease. Inside a tumor, the linker is snipped, releasing a deadly toxin directly into the cancer cell, leaving healthy tissue untouched. Linkers are not merely biological adhesive. They are programmable triggers that give engineered drugs spatial awareness and targeted precision. The architectural layout of the protein is also critical. Placing a domain on the N-terminus, the front of the chain, versus the C-terminus, the back, dictates the drug's stability and how much the cell produces. As illustrated in panel A, placing a protein domain backward can lead to total misfolding or a complete loss of potency, requiring scientists to test multiple orientations to find the one that works. Once the layout is optimized, engineers can use oligomerization. This involves stacking multiple fusion proteins together to increase their avidity, or their total grip strength on a cellular target. By utilizing self-assembling domains like p53 or antibody Fc regions, scientists can scale molecules from simple dimers up to the complex hexameric structures seen here on the far right. Manipulating this spatial geometry transforms a single binding molecule into a multi-armed biological grappling hook. To address the issue of rapid kidney filtration, engineers utilize specific structural building blocks: human serum albumin and the antibody Fc domain. These domains are engineered to hijack the body's internal recycling pathway. When a cell internalizes the protein, it enters a lysosome, an acidic environment usually meant for waste disposal. However, inside this acidic bubble, the protein binds to a rescue mechanism called the FcRn receptor. While other proteins are broken down by enzymes, the FcRn receptor shields the fusion protein, protecting it from destruction. The receptor then ferries the protein back to the cell surface and releases it back into the bloodstream at a neutral pH. This recycling mechanism can extend a drug's lifespan from mere minutes to several weeks, significantly reducing the frequency of injections for the patient. While the design of fusion proteins is mathematically precise, physically manufacturing molecules that do not exist in nature is a significant challenge. When grown inside microbial hosts like E. coli, these unnatural proteins often fail to fold correctly, collapsing into dense, useless clumps called inclusion bodies. Producing a functional drug from these clumps requires a complex industrial process. As shown in this microbial bioprocess flow, the cells must be disrupted and the inclusion bodies isolated before scientists use chemicals to force the proteins to refold into their active shapes. Even with successful folding, there is the risk of immunogenicity. the human immune system may identify the artificial junctions between the two proteins as alien, causing it to attack and neutralize the medicine. Despite the manufacturing difficulties and immune risks, fusion proteins demonstrate our ability to reorganize biological building blocks into targeted, life-saving medicines that